Hello, my name is Bernhard Hauer. I'm from the Institute of Technical Biochemistry at the University of Stuttgart. In this webinar, I will talk to you about our work on Squalene Hopene Cyclases. This work was funded from the European Union under the 7th Framework Program, and the name of the project was Kyrobio. First, a few words about our institute. It's located at the University of Stuttgart in the southern part of Germany, and we are a rather interdisciplinary group of researchers coming from chemistry, biology, and a few other disciplines. And we do biocatalysis, but we also work on functional materials and do bioinformatics. Currently, we are about 35 PhDs and postdocs, and the people involved in this project I'm talking to you are Miriam Seitz, Silke Bastian, Jörg Domenikus, Stefan Hammer, and Bettina Nestel. The general theme and goal of our group is to expand the range of accessible reactions with novel enzymes and thereby offer new biosynthetic routes to target compounds. Contributions are necessary from many disciplines, including chemistry, biochemistry, but also molecular biology, microbiology, bioinformatics, and bioengineering. If you are interested in other things we're doing, you can also always have a look at our homepage, and access is located in the lower part of this slide. So the general question, as I said before, is that we are interested in novel biocatalytic functions, and we want to catalyze non-physiological reactions, things which are not so known in nature, and the question is how can we identify and develop these kind of enzymes. And we can always try very classical things like doing microbiology, screening databases, or you go to the other extreme and do computational de novo design. Both things are not in the center of our focus. We rather do a more chemistry-based approach and look at the mechanism of existing enzymes and try to exploit them for new reactions. This is what you see in the center of this slide. You see the native substrate sort of fits into the catalytic site, and then we try to find promiscuous substrates which are also fitting in the active site and are catalyzed in a new way, and thereby a new reaction is performed. One of these examples for new reactions is done with the enzyme called Squalene Hopene Cyclase. This is an enzyme which is doing the cyclization of squalenes. You see that at the upper part of the slide here. And this squalene is protonated at one end of the molecule, and it's pre-folded in the active site, and then in one reaction the several bonds are formed, several chirality centers are formed, and at the end of this reaction you have either quenching by a water or by a base, and hopane or hopanol are formed. These are compounds which are necessary in the microbial membranes, and this is why you find a lot of these enzymes in microorganisms. These enzymes are associated to the membrane, and they are usually dimers. You see that here in the lower left part, where these enzymes in this cartoon should give you an impression how these enzymes are attached to the membranes. And this is important. I will come back to that later. Then you see in the center monomer, and there it's already a little bit visible that the active site is really a deep pocket where these long terpenes can access and are pre-folded. And at the end, and now we go to the right part, you see at the end is a catalytic amino acid. It's an aspartate usually, and this is donating the proton to initiate the entire reactions, where carbocations are formed, and then they sort of zip through the molecule, and the quenching at the end takes part. Now usually an aspartic acid is not so acidic to perform such a reaction, such a protonation reaction, and the question is how is that done? And we find here a very particular and interesting situation. And you see that on the right again. Usually aspartates are in a so-called syn orientation, where you see the oxygen, the ketone, and the hydroxy group, and the hydrogen are on the same side, so to say. If they go into this anti-orientation, where you have the proton opposite to the oxygen here, the proton is calculated to be about 1,000 times more acidic than in this syn orientation. And so we have like a special catalytic site in these enzymes, and you see that nature really takes a lot of efforts to make that possible. So there are other amino acids involved in a little network to create this anti-orientation. The first thing we did with these Squalene Hopene Cyclases, which we can also call a proteinase if you want, is that we looked at the substrate scope, and we looked at different chain lengths, and you see here quite a set of substrates from C30 to C13 down to C13, and all these substrates are converted. So there's a certain flexibility in the chain lengths, but if you look on the color part, on the terminal part of the molecules, you see that they also accept different functional groups at the termination of the reaction, or the way this reaction is terminated. So if you have a ketone, for example, here, you form an enol ether. If you have an acid, you form a lactone, and if you have an alcohol, ethers are formed. So you can create quite an array of heterocyclic compounds, and you can convert not only these very long terpenes like C30 molecules, but you can also go down to C13 to rather small molecules. We even went a bit further and did not unmodify these molecules even further. You see that in this very busy cartoon here. So again, when we have as an X here, we can, which is an alcohol, for example, we can form compounds like ambroxan, which are fragrance compounds. We can even have phenolic compounds as X. We can have the carboxylic acids, ketones, and aromatic rings. And if you look, for example, on this compound with the aromatic ring, you see that we have a bond formation which is something which we call a Friedel-Crafts reaction. So we get an alkylation of this benzene ring here. But we can also have nitrogens in our precursor molecules, for example an amide, and then heterocycle is formed at the decahydroquinoline core, which you find in various alkaloids. Also thiols are possible as starting materials, and then we end up with a sulfur component. All these different compounds here are very interesting because they're very similar to nature materials or compounds you find in nature. I mentioned already the ambrox flavor compounds. Sclariolit is another compound interesting in the fragrance industry, but we can also come close to anti-cancer compounds or totarol compounds, which are antibacterial compounds. We have one disadvantage, however, that really small molecules like C10 terpenes are not converted so far by this enzyme. Before I will continue with looking at new reactions, let me say a few words on the reaction itself. As I mentioned before, this enzyme is associated with the membrane, and so it's not so easy to work with it, but also not too complicated. You should not be worried, but we always have to add a little bit of a surfactant or detergent to the reaction to keep the system running. And we were working with two compounds, tritone or tritone X100 and JAPS, which is a charged surfactants. And when we now look on some of the reactions, we can first have a look at the left side here, where squalane is converted to these hopenes and hopanols. That's the natural reaction. And you see, if we run the reaction in the presence of tritone X100, we get product formation in the dependence of detergent concentration. These are data which we collected after 1 hour reaction time. If you run the same reaction in CHUPS, this is in blue now, you see there's hardly any conversion. You would not discover the reaction product. If we now go to a not so natural product, homofarnesol, which is close, the ring is close to the compound ambroxan, you see there that CHUPS as well as tritone are working, but you see the less detergent we add, the better it is. And when you go to higher concentration, tritone is a little bit less suitable for this type of reaction. If we now go a bit further with this and look at 2 other reactions and 2 different enzymes, the things are even more surprising. This time we look on the ring formation based on geranyl acetone to this heterocycle here, and we study the temperature dependence with 2 different enzymes now. One from Cymomonas, this is a bacterium, a gram-negative bacterium. The enzyme is said to be mesophilic, and we study the enzyme from another bacterium, Alocyclobacter, which is said to be thermophilic. And again, we study both detergents, and at the left, see both are, let's say, very similar. We have a temperature optimum here for the Cymomonas enzyme around 40 degrees. If we now go to the right to the Alocyclobacter enzyme, with the big difference between the 2 detergents, CHUPS is giving us quite some activity already at 30 and 40 degrees Celsius, whereas tritone is rather low, the activity, but then the highest activity is around 55 degrees Celsius. So the enzyme works better at higher temperatures, but overall we get more conversions if we use CHUPS at a lower temperature. So the detergents behave very similar, and so far we can only say that we have to study these detergents very carefully with these substrate, with these enzymes, but so far we have no complete understanding of this all. Now I would like to come back to finding and new reactions. As I said before, substrates like farnesol, neroldiol are accepted. We can make different compounds out of these molecules, and but there are also compounds which are not accepted. For example, geraniol, a small terpene, is not accepted as a substrate. Also linalol is not accepted. Pseudoionone also not accepted. These reactions on the right side would be of quite some interest, because they all would lead to interesting compounds, either to compounds which you can use, for example, in polymer chemistry. You can see there are diols which are bridged. You even might have a possibility to do some cross-linking with these molecules. Other molecules like this one based on citronellal would be a precursor for L-menthol. This is a very important flavor compound, but you see some other fragrances compounds or ethers and lactones here on the right side, just to demonstrate that it would be really very useful if we can do the monocyclization of these monoterpenes. They are very difficult to make in chemistry. This is shown on this slide. With an enzyme we would have the option to do that in one step, and as you see here, to really come to a clean chiral product, it's a very laborious 6-step total synthesis, and it's really not so easy. I think that's obvious by just looking at these slides. You generate also a lot of waste material. You have to handle reagents which are not so easy to handle, and you have to work at rather low temperature. So it's really quite an effort to synthesize this compound, whereas with our cyclases we could probably perform, if they would accept these molecules, in one step. So how to develop such an enzyme. So the first thing we asked is, are these small substrates entering the active site? Can they really form as a template for the reaction? And up there you see again a cartoon of the active site with the active amino acid with the aspartate and the pre-folded terpene in the pocket. Now what we did is first we did some simulation work on the computer, and we found that actually these small substrates would fit into the pocket. They could also fold into the right way. So this is not a problem, but when we really put them into the position, what we see is that they don't stay there. They move out of the active site, they unfold and sort of leave the active site again. So we get no near attack conformation, no cyclization is taking place. And these studies were confirmed with NMR studies, which also pointed to us that the substrate can enter the active site, but we get no near attack conformation, so conversion is not taking place. So we need a special binding for these enzymes. So our idea was, can we not modify the active site to allow this near attack conformation? So we started to make a library of the active site, to reshape the active site, to improve the productive binding of these non-natural substrates. And here on the left, some more details are given. Now you see a small molecule here. As you see again the active amino acid here, and then you see other amino acids which sort of cover this active site, which really make this pocket. And what we said, we don't touch, as our strategy, we don't touch the first shell of amino acids which make the catalytic machinery. So 8 amino acids were excluded. The next step was that we said we want to retain the hydrophobic character of the active site, which meant we didn't exchange the amino acid to any amino acids. We exchanged them only to other hydrophobic amino acids like alanine, phenylalanine, tryptophan. And we also made a few additional double and triple mutants when we saw beneficial effects of single amino acid changes. So with this strategy, we designed about 50 variants of the cyclases and tested them against 3 substrates, small molecules like geraniol, oxidogeraniol, and citronellal. You see they are all small, but you see also that we have now different functionalities at the protonation site. We have this usual terpene motif in geraniol. We have an epoxide in the 2,3-oxidogeraniol, and we have actually an aldehyde in the case of citronellal. And these molecules were now tested with what the library was screened with these molecules, and we identified quite a few mutants which now converted these substrate into single products, and they were all these reactions were done enantioselective, selective, and I will now show you what the results are. So with geraniol we found actually 2 mutants, the F365C and the G600F. You see again that the wild type converted almost nothing of these substrates, and now the mutants, in one case the conversion was 15%, and with the second mutant 68%. And you see the reaction was really selective. The EE value was higher than 99%. Also oxidogeraniol could be converted now with a different set of mutants. It was a double mutant or a single mutant. The wild type, again the conversion pretty low. We saw with the single mutant about 54% conversion, with the double mutant 78% conversion. The product very clean, 1 compound and more than 99% selectivity. With citronellal, actually the S form was the substrate. We also got one product here and 11% conversion, more than 99% selectivity. So it's really interesting that by reshaping the active site, we could convert all three molecules with a very special mutant to a single product. And there was also no cross activity. The citronellal mutant did not convert oxidogeraniol, or the geraniol mutant was no conversion with the oxidogeraniol. So all mutants were very specific for their substrates. I think that we have shown that we can not only do polycyclization with these enzymes, we see that we can do monocyclizations. They open to us access to rather new products, and at the moment we try to go even further and use the powerful biological protonation machinery as a platform for Brensted acid catalysis. And in principle, these enzymes should be able to do Friedel-Crafts reactions. You have seen one little example for already one Friedel-Crafts reaction, but reactions of the type shown here are more general and would be much more looked for. Also, pinacol arrangements should be possible, Michael additions, Prince-Ehn reactions, Nassaro cyclization. And what we have learned so far is that the enzyme and the reshaping of the enzyme is one thing, but we have also to be very careful in selecting the substrates, that they not only fit into the active site, but they fit into the active site in a pre-folded way to make the reaction possible. And this is very important when we design the substrates, that these substrates can really move, rotate into this pre-folded active position. So I hope the next time I can tell you more about these Brensted acid catalysis possible with the Squalene Hopene Cyclases, and thereby we can add another tree to these functions and enzymes doing cyclase reactions. At the end, I like to acknowledge again the people in the group. I mentioned all the names, and of course Kyrobio for funding. And if you have further questions, don't hesitate to send us an email, and I hope we have the right answer for you. With that, goodbye and see you soon.